Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Serial dilution methods. Understanding how to estimate cell concentration in cultures. Table of contents. Introduction to serial dilution. Serial dilution objectives. Serial dilution formula and calculations. Online serial dilution calculator. Procedure of serial dilution. Applications of serial dilution. Limitations of serial dilution. Serial dilution examples. Introduction to serial dilution. Serial dilution involves performing a series of sequential dilutions to reduce the concentration of a dense solution to a more manageable level. In other words, it is the process of gradually diluting a solution in steps, each with a defined dilution factor. In biology, this technique is commonly used to lower the concentration of cells in a culture, making them easier to work with. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Serial dilution objectives. The primary goal of serial dilution is to estimate the concentration of organisms, such as bacteria, viruses, or colonies, in an unknown sample by counting the number of colonies cultured from various dilutions of that sample. With each step in the serial dilution process, the cell density decreases, making it easier to calculate the original concentration by determining the cumulative dilution across the series. Serial dilutions are often used to avoid pipetting extremely small volumes, 1 to 10 microliters, when preparing a solution. By diluting a sample in a controlled manner, this method allows for culture plates to produce a manageable number of colonies, typically between 30 and 100, enabling accurate counting and calculation of the microbial load in the sample. Serial dilution formula and calculations. Serial dilution is the process of progressively diluting a sample using standard volumes of a sterile diluent, typically distilled water or 0.9% saline. A small measured volume from each dilution is then used to prepare a series of pour or spread plates. The extent of dilution is based on the estimated concentration of cells or organisms in the sample. For example, a highly polluted water sample requires a higher dilution factor, whereas a lower dilution factor may suffice for less contaminated samples. Serial dilutions, often in 2-fold or 10-fold steps, are commonly used to titer antibodies or prepare diluted analytes in laboratory settings. The dilution factor for a serial dilution can be calculated for each individual test tube, or as a cumulative total for the entire series. Dilution factor for each tube in a series. In a 10-fold dilution, for instance, if 1 milliliter of sample is added to 9 milliliters of diluent, the dilution factor for that tube will be, after the first tube, each subsequent tube is a dilution of the previous one. To calculate the total dilution factor for the second tube equals dilution factor of the first tube, multiplied by dilution factor of the second tube. Example: for the first tube, the dilution factor equals to 10 to the power of 1. 1 milliliter sample added to 9 milliliters diluent. For the second tube, the dilution factor equals to 10 to the power of 1. 1 milliliter sample added to 9 milliliters diluent. The total dilution factor equals to dilution of the previous tube. Multiply by dilution of the next tube equals to 10 to the power of 1. Multiply by 10 to the power of 1 equals to 10 to the power of 1. Dusra. Online serial dilution calculator. Here are several online tools available for calculating serial dilutions. AAT BioQuest, Serial Dilution Calculator, Merck, Solution Dilution Calculator, Omni Calculator, Serial Dilution Tool, End Memo, Serial Dilution Calculator, HandyMath, Serial Dilution Calculator, Tocris Bioscience, Dilution Calculator, Physiology Web, Dilution Calculator, Mass per Volume, Celic Chemicals, Dilution Calculator, Apex Biotechnology, Dilution Calculator, Cite-AB, Dilution Toolbox, and Fluffy Frog, Dilution Calculator. These tools provide quick and easy solutions for performing serial dilution calculations online. Procedure of serial dilution. The following is the procedure for performing a 10-fold serial dilution to achieve a final dilution factor of 10 to the power of 6. Begin by taking the sample or culture in a test tube, along with 6 additional test tubes, each containing 9 milliliters of sterile diluent, distilled water, or 0.9% saline. Use a sterile pipette and draw 1 milliliter of the well-mixed sample or culture. Transfer this 1 milliliter into the first tube, bringing the total volume to 10 milliliters. This results in an initial dilution factor of 10 to the power of 1. Thoroughly mix the contents of the first tube by repeatedly filling and emptying the pipette. Discard the pipette tip, attach a new sterile tip, and draw 1 milliliter from the first dilution, 10 to the power of 1. Add this 1 milliliter to the second tube, resulting in a total dilution factor of 10 to the power of 2. Repeat this process for each subsequent tube, transferring 1 milliliter from the previous dilution into the next 9 milliliters of diluent. After completing all 6 tubes, the final dilution for the sample will be 10 to the power of 6, 1 in 1 million. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Applications of serial dilution. Serial dilution is widely used across various experimental sciences, including biochemistry, pharmacology, physics, and homeopathy. In microbiology, it is employed to estimate the concentration or number of cells or organisms in a sample, allowing for the preparation of an incubated plate with a manageable number of colonies for counting. In biochemistry, serial dilution is used to achieve the desired concentration of reagents and chemicals from a higher starting concentration. In pharmaceutical laboratories, it is performed to obtain the required concentration of chemicals and compounds, as this method is more efficient than making individual dilutions. In homeopathy, serial dilution involves diluting a substance in distilled water or alcohol. It is believed that the dilution process enhances the substance's potency by activating its vital energy. Limitations of serial dilution. Despite its usefulness in laboratory settings, serial dilution has several limitations. Errors can occur during the sample propagation process, and transfer inaccuracies can lead to reduced precision and accuracy. The highest dilution is particularly prone to these errors, resulting in the least accurate outcome. Since serial dilution is performed in a stepwise manner, it can be time-consuming, which limits the method's overall efficiency. While serial dilution reduces the concentration of bacteria or cells, it does not separate them, unlike techniques such as flow cytometry. Additionally, this method requires skilled microbiologists and experts trained in aseptic techniques to ensure proper execution. Serial dilution examples. Examples of serial dilution can be found in everyday life and scientific applications. A common example is preparing tea or coffee. When making coffee, we dilute a concentrated amount of cold-pressed coffee with water to achieve the desired strength. In chemistry, serial dilution is used to dilute acids and bases to obtain a specific concentration. In microbiology, a key example is the serial dilution of a culture to determine the number of bacteria in a sample using a plating technique. This allows for an accurate count of bacterial colonies. Thank you so much for watching. Don't forget to subscribe to my channel. Press the bell icon button. Thank you!